So our fourth speaker, Tristan, if you want to go ahead and bring up your slides, is Tristan Durand, who is coming to us today from San Diego at the Scripps Institution of Oceanography. And so Tristan first gained an appreciation for metabolism working with Streptomyces bacteria as an undergrad at Brown, where he was working with Jason Sello. And then from there, he went on and joined Jay Kiesling's group, so a connection with our third speaker, Ryan, in the Kiesling group at Berkeley, where he focused on enzyme discovery using high-throughput mass spectrometry. And now he's an NIH postdoctoral fellow working in Brad Moore's group, where he's looking at exploring what the world's oceans have to contribute to the biosynthetic toolbox. And so I think we're in for an interesting talk today from Tristan. Are you ready to go?
I'm ready, yeah.
All right, take it away.
Thank you very much for having me, Allison. And it's fun that Brad just mentioned the idea of enzymes that do multiple reactions in a kind of synergetic way to carry out one overall transformation. That's going to be one of the big themes of this talk. So first, I'd like to thank Allison and the other organizers of this little symposium for having me. I think you guys did a really great job kind of selecting speakers from all kinds of different steps along the pipeline of the biocatalyst field. So, you know, this is kind of what I'm getting away from today's day. You know, we start with enzymes that we discover somehow, and we're engineering them, we're formulating them, planning them into biocatalytic cascades, and then at the end, maybe we can do chemistry as cool as Merck is doing, right? And so the topic that I would like to talk about right now, you guys can see my arrow here. I'm going to change it into the pointer here, is these enzymes. You know, we can improve enzymes all we want, but we got to get the enzyme from somewhere in the first place, right? And I see that as my job right now to try to discover these new fundamental classes of enzymes that we're going to be using to do cool biocatalytic chemistry. And, you know, people have different ways of coming about these enzymes. Our group has done a lot of reimagining where they, while they're engineering something, they realize an enzyme can do something interesting, and then they really focus until it's almost no longer the parent anymore, right? Some people are doing the novel enzyme design, but I think that field still has a bit of a way to go. But I think the majority of enzymes, you know, we're getting from metabolic studies where somebody has noticed an interesting metabolic pathway, and they're like, oh, look at that transformation, you know, maybe we should develop that enzyme into a but I want to kind of talk a little bit about a different approach. You know, if we look at nature, and this is the view we get from our office at SAO. Unfortunately, you know, I haven't actually been there in over a month. So as good as a view it is, unfortunately now I don't use it very much. But if we look at nature, you know, there's probably a lot of enzymes out there, right? Like nature has been evolving for billions of years to all kinds of cool chemistry to try to kill each other, to try to eat each other. You know, I imagine that there must be some really cool enzymes that have evolved in there that we haven't noticed yet. And we're getting really good at sequencing, right? So we can just sequence lots of DNA, and then we should learn something from all that nature, right? But it's not that easy because, you know, you sequence something, you assemble it, and then when you look at the results, you know, the majority is hypothetical protein. So, you know, just because you sequence something doesn't mean you have insight, right? You need to actually make some selections, do some testing, and then maybe you can find new biocatalysts. And while you're at it, you know, maybe you can improve the databases for the people who are doing functional phenotyping and help them along the way as well. So this field of taking genome information and trying to make selections and then trying to find, well, traditionally people would be finding bioactive molecules, but you can imagine you can do it for catalysts as well. It's called genome mining. The most common way to search for hypothetical proteins that you might want to look at is to look at proteins that are in metabolic gene clusters that have some similarity to known bioactive metabolism. So maybe bulky synthases, non-ribosomal peptide synthesis, right? Those are very well studied pathways, and we know how to recognize them, and we use tools like anti-smash, PRISM, to find those kind of metabolic pathways, and then maybe we prioritize them, and then either do heterologous expression or knockouts or something to figure out what they do. But almost every pipeline is kind of premised on this, you got to find the cluster first. And if you're not looking for new things, then you're going to just be finding things that we've already seen before, right? Like if you're searching for PKSs, you're probably going to be mostly finding PKSs. And while PKSs are really interesting, maybe if we want to find fundamentally new chemistry, it might be a little bit more difficult to do it that way. So how do we find fundamentally new enzymes? And I'd like to just do a little thought experiment with you guys. So imagine my mom sends me a text. Looking forward to Mexican food tonight. I hope you'll surprise me. My mom is very picky. She really wants to be surprised. I don't want her to eat anything that she's had before. And of course, I thought about this, right? Oh no, it's tonight and I haven't thought about it yet. Why don't I pull up one of these apps where, I search for the ingredients, and maybe it will tell me what kind of recipe I can make. So, I go through cupboards and like Mexican ingredients, tortillas, chili peppers, right? Tell me what's in there. And it comes up with thousands of results because, lots of things in Mexican food have tortillas and chili peppers, right? So tacos, burritos, nachos, and all that food is delicious, right? But my mom has probably already had it, right? And I'm not saying that there isn't a burrito out there that has something new going on, like, maybe cactus in it or something like that. But if you have to go through those thousands of recipes, maybe you're not going to find the novelty right away. So I look through my cupboard. I'm like, oh, actually, we have some chocolate in the cupboard as well. Maybe, I don't know, maybe let's search that. It turns out, oh yeah, there are dishes with chocolate and chili pepper, right? Maybe there's mole. If any of you haven't had mole, it is absolutely delicious. It's very, very different from anything you've tasted before. So how do we find the mole of enzymes, so to say? And I can even quickly send my mom a message, without even spoiling what I'm going to make her. Just asking, have you ever had a dish with chili pepper and chocolate? And she's, no, I've never had that. So that's pretty exciting. All right. So I think of these ingredients as domains in enzymes. All right. And the majority of enzymes we talked about today have single domains. So for instance, the cyclohexylamine oxidase has a flavin containing amino oxoreductase domain. People define their domains a little bit differently depending on which organization you're with. But, generally one domain is doing one kind of reaction. We'll get into that in a bit. Ryan's talked about some ketoreductases, some transaminases. These are all really cool catalysts. But I do kind of think of it as eating a single ingredient. Maybe you're only having a tomato or only having a cucumber, which is absolutely delicious, but we can do better, right? Maybe let's think about non-ribosomal peptide syntheses. So this was a great animation that was made by Michael Maloney that shows out how a bunch of domains are working together to do chemistry that any of the individual domains wouldn't be able to do, right? The ATP binding domain is adenylating an amino acid. The condensation domain is condensing it with the next enzyme over. And these types of NRPSs are very well studied. And if you search for, the AMP binding domains and the condensation domains, you're going to find NRPSs, no question about that. That's how a lot of those tools work. But there's a lot of NRPSs out there that do very similar work, and sometimes they're going to have a cyclase domain, right? Which is a little more exciting, or sometimes they're going to have a different domain that's even more exciting. But I do kind of think of these as like the beans and rice of domains. We're going to find more of the same if we just search for these. So, like I said, how do we find? Oh yeah, this is kind of like the burrito of enzymes, so to say, right? So how do we find the mole of enzymes, right? Last year, there was an interesting report from the Bauskas lab of a didomain enzyme that does this really cool oxidative rearrangement of a methyl arginine to form this N-nitroso compound over here. how do we find enzymes like that? I think that that's really cool. So can we do that in a more systematic way? How do we search for, the weird domains that we haven't spotted together before, to like go through all the databases, and how do we find those particular examples? So I'm going to be searching for the co-occurrence of enzyme domains, and that makes a nice abbreviation, co-ed, which also kind of evokes the idea of, co-education, right? Like back in the days, people would only have men's schools and women's schools, but now we have co-educational schools, and things are more interesting, and people are learning better in those schools because they can work together. All right, so let's look at how these results would work. I wrote a little script, and it goes through a genome and basically annotates the domains and which ones are found together. So here's an example of the E. coli genome and all the proteins that are multi-domain in E. coli, and which domains are found together. And you might notice, for instance, here's an AMP binding domain and a colonization domain, as you saw before in an NRPS. So maybe this is an NRPS. And as we know, E. coli has an NRPS that encodes enterobactin. So that at least it picks that one out, right? And maybe here you have a transpeptidase, transglycosidase, pair of domains. We know those are the penicillin binding proteins. But it might be a little bit easier to look through these domains. I can hover over them and get a little bit more information about what the domains truly are. But I want to annotate a little bit. The way that I texted my mom and asked, "Have you ever had a dish with chocolate and chili pepper?" I want to ask the databases. And interestingly, as Will talked about earlier today, not every database has every piece of information. So I had to kind of combine a couple of different databases, Brenda, Uniprot, MyBig, and kind of figure out which combinations of domains have been annotated before. And sometimes they're in all the databases, but sometimes they're only in one or a couple. So the enterobactin, like NRPSs, have been annotated in all of these databases, or to be more accurately, that combination of domains has been annotated in that database. And in fact, every single edge in this network over here is color, which means that E. coli is probably not the best place to be mining for multi-domain biocatalysts anymore. But maybe let's look at some slightly less model organisms. Here is Streptomyces color. We can immediately see that it has a bunch of megasynthases because it has a lot of combinations of domains that are in this big blob together over here. And it has a lot of enzymes that have some kind of annotation. But there's also a lot that are not. Here, every arrow that's gray over here is something where that combination of domains has not been seen together in a database before. So maybe we could find a couple cool biocatalysts here. I decided to go into this organism, Pseudoultramonas rubra, which is an organism that I worked on in my graduate work. When I was working on it, I was interested in this reaction here, the cyclization, oxidative cyclization of proteoglycan, and this was being catalyzed by a 9-transmembrane protein, and I thought I was going to have trouble heterologously expressing in E. coli, but it just worked on my first try. So I'm like, heterologous expression from this organism works well, so why don't we pick an enzyme from this organism to study further? So when I looked at this co-ed network, I noticed here the combination of a Thi-F and a nitroreductase domain. The edge between the two is gray, so those two have not been annotated together in a database before. And those domains stood out to me because they are known to do pretty interesting chemistry. The Thi-F domain is known to be quite important in primary metabolism. It helps make cofactors such as thiamine. It is also involved in the ubiquitin activase chemistry that is part of the ubiquitination pathway of protein degradation. The nitroreductase family domain does a lot of really cool chemistry. Of course, it does nitroreduction, it does desaturation, it does oxidative ring contraction, it does reductive dehalogenation. So I'm like, I've never seen these two domains together, but they're both doing cool chemistry. So maybe they'll do cool chemistry together, right? And in our organism, in Pseudoultramonas rubra, this is next to an azyltransferase. So maybe that's important. Another cool thing about this tool here is the numbers here. For instance, the bottom number is how often this pair of domains shows up in UniProt, not just annotated, but in any capacity. So it shows up once in our organism. Let me zoom in a little bit to make sure people can see it. It shows up once in Pseudo-Ultramonas rubra, but it shows up 50 odd times in UniProt, and I can get all those UniProt identifiers. I can query UniProt against it. I can put it into the genome neighborhood tool and figure out what's next to it and get all kinds of interesting statistics about these types of enzymes. And so what I learned is that this enzyme is found throughout alpha proteobacteria, throughout proteobacteria in general, alpha, beta, and gamma proteobacteria, mostly found in aqueous environments, mostly marine, which is to my heart, right? My office is right next to the ocean. And that N-acyltransferase is usually next to this enzyme. So I call them OXA, OXB. All right, so at this point, let's do some heterologous expression. I really like these kind of experiments where I just take some genomic DNA, PCR it, heterologously express. Conceptually, it's really simple. I love to take on undergrads who can help me with these kind of experiments. They can learn how to PCR, they can learn how to clone, they can learn how to do heterologous expression, then do the analytical chemistry and figure out what they're making. So when we heterologously express the OXCAB gene cluster, our E. coli colonies turned yellow. Well, that's great. There is a phenotype, right? This enzyme is doing something interesting. And when it's making something yellow, that makes it easy to purify, right? So we can do UV HPLC and see that it's making a variety of yellow compounds. And after many, many weeks of purification, or months even, and NMR analysis, especially guided by adequate and nitrogen HMBC experiments, we figured out that these are the structures that are being made. And these are new compounds that have not been found in the literature before, and they are of a class called oxazolones. So oxazolones, kind of a big word, right? So the ox is an oxygen, the az is a nitrogen, the one is a ketone, and then the whole thing together is a heterocycle 5-membered that has all of those features. And oxazolones are not very common in the natural product literature, and they're found a little bit, but I think they might be really interesting synthetic intermediates for biocatalytic cascade, for instance. So we have a lot of electrophilic positions. We have pi systems that we might be able to do cycloadditions into. We have some nucleophilic systems. So there's a lot of interesting chemistry you can do with these kind of molecules. And I think we might be able to come up with some cool chemical cascades that take advantage of them. So in nature, they are found a little bit. There's a couple chalcophores, the methanobactins, that have oxazolones. The Basenbeck genes are known, and they're totally different from the ones that I found. And they act on peptides, so it's a little bit difficult to use them as biocatalysts. There's one more class of oxazolones known that was found in a seaweed, and nobody knows the genome or how these are made. If I were to guess, it looks like an amino acid. It's probably a cluster that's very similar to the OXCAB that I discovered. So the ones that look like tyrosine, I like to call them tyrazolones, tyrosine-derived oxazolones. You can imagine that the N-acyltransferase probably takes tyrosine and acylates it, and then this new OXCB enzyme probably turns that into a oxazolone. You can imagine it has an oxidative domain, it has an ATP binding domain, so together they might be able to do a cyclization, oxidation, and then we're in the right oxidation state for the product. So let's see if that works in vitro. If we purify the N-acyltransferase, OXA, and incubate with tyrosine, it does in fact make N-acyltyrosine, so that's great. We can synthesize N-acyltyrosine and incubate it with the OXCB enzyme, and in the presence of ATP, we see the oxazolone product. So this is the first example of a enzyme that's been proven to be an oxazolone synthase. And if you put the very beginning starting materials together with all the enzymes, then we also make the oxazolone. So that's exciting. It's an oxazolone synthase, and it works in vitro. It also works in vivo. I know how much some of the biocatalysis people like doing these kind of reactions in vivo. You can put OCB into E. coli into a big flask, and as long as you aerate it well enough, it will catalyze this transformation. And as you might notice, the oxazolones, specifically the ones derived from tyrosine, are in a cis-trans equilibrium between the two, and you can see that because there's always two peaks. And let's look at substrate scope, because I think a lot of the biocatalysis people care about that a lot. So the pseudo-ultramonas enzyme, you know, from the in vivo and in vitro reactions just now, we know that clearly it takes an N-acyl tyrosine as a substrate. And we decided, you know, let's give it some other substrates, some different aromatic systems, some non-aromatic systems. And it turns out, you know, only if it's very, very similar to the native substrate, so to say, does it work maybe a little bit. This was a very small peak. So I think we need to do better. We need to find a better oxazolone synthase for biocatalytic purposes. And we can either try evolving it, but remember, you know, when I was looking through UniProt, there is another 50 odd examples of this enzyme. You know, maybe there is, maybe we can find one that has better substrate specificity that we can take advantage of. So I took one more enzyme from an alpha protobacterium, one more from a beta bacterium, and two more from a gamma protobacteria, and tested what they made in vivo. So in P. rubra, we now know that it makes a bunch of tyrazolones, right? And the OXEB from all these, or OXEAB together from all these other organisms, they all make the tyrazolones, which I think from a functional gene annotation perspective is great. You know, at least they all do the same reaction, so we can, in the future, when we see an enzyme like this, annotate what it does. But some of them make other peaks in addition to that, right? So for instance, here is a peak. And after a little bit of purification, we figured out that those are the phenazolones. These are kind of similar, but are derived from a phenolphthalein instead of a tyrosine. These actually don't isomerase in our hands. So probably interesting tautomeric situations going on over there. So if we take one of the enzymes that makes both phenazolones and tyrazolones, maybe that will have a little bit more relaxed substrate specificity, right? So previously, the P. rubra enzyme doesn't really act on much of anything. This other oxy homolog acts on a lot of different substrates, but maybe not if it changes too much. You know, I think the tryptophan-derived ones, I would not be surprised if at some point in the future in nature we'll find one that can accept something like this. But it can accept, you know, different aromatic systems, quite dramatically different aromatic systems, and it will even accept non-aromatic substrates. So I think there is a lot of potential for interesting reactions that we might be able to do this with this. And of course, you know, there's a lot more evolution we can do on these kinds of enzymes. So in short, you know, I think co-ed can help us guide finding new biocatalysts. I just want to point out here, if we look at the co-ed network of all proteins in UniProt, this is way overwhelming. But we can kind of see how many of domain pairs have ever been annotated. And if they're proteins that are really common in life, I think we've done a pretty good job annotating them. But if they're proteins that only appear, you know, 100 times, and 100 times is still quite often, you know, the majority of domain pairs have not been annotated yet. And if they only appear 10 or more times, the vast majority, there's thousands more pairs of domains that have not been annotated before. And the oxazolone synthase that I just talked about has about 50 examples in UniProt, right? So there might be thousands more examples of cool biocatalysts. And some of them might not be, right? Some of them might just be two primary metabolism enzymes that happen to have fused. But some of them will be cool new biocatalysts. So I think this can help us prioritize those kind of information. And using these kind of tools, we can help find all those, right? In Cytoscape here, we can filter for anything that has not been annotated, anything that has more than 1,000 members. Let's say we're interested in functional gene annotation. And then we filter for them, and then we get a network like this one, where if you wanted to find all combinations of domains that have more than 1,000 different examples in UniProt, here's all of them. Maybe we want to start digging through these. Maybe these are important enzymes for biocatalysis, but also for all kinds of interesting biochemical questions. All right. So that's co-ed. It can help us find new catalysts. We have specifically found biocatalysts that can make oxazolones. And I would love to know what you might want to use oxazolones for. You know, if you want me to try out a particular substrate because you're interested in a particular product that you might want to use for a cascade or something like that, let me know. I would also love to know how you'd like to use co-ed. You know, I wrote the code right now as part of a Jupyter notebook, but would you rather have it as a command line tool? Would you rather have it as a web server? You know, if people are interested in these kind of tools, tell me what features you want and what kind of format you'd like it in, and maybe I can make it available for as many people as possible. And this work will be published, or will be a preprint on it will be published very soon in the code as well. So keep an eye on maybe my Twitter or my website, and I will make that available very soon. So with that, I would like to thank the Moore Lab, the NIH, and specifically a lot of the undergrads I've mentored over the last couple of years. It's a little bittersweet right now that a lot of them are graduating, but I am not even able to celebrate that with them in person. You know, we still have weekly little subgroup meetings where we discuss papers. Just the other day, we were discussing the CRISPR-Cas9 paper, and it turns out Cas9 has two catalytic domains. You know, one to cut the top strand, one to cut the bottom strand. If those two catalytic domains weren't working together, then it wouldn't do a proper double-stranded break. So Tristan did a lot of the substrate specificity work, helped me synthesize some of these substrates. Julia was instrumental in the original cloning and purification and structural elucidation of these classes of enzymes, and Michelle will still be working on some of the biochemical questions that we're asking about these enzymes. So thank you very much for your attention, and I would be happy to answer any questions.
Thanks for a fantastic talk, Tristan, and also for a set of questions to guide the discussion. We already have one hand up. Mitchell Thompson, do you want to go ahead? You should be able to talk.
Hey, Tristan, can you hear me?
Yeah, I can hear you.
Great talk as always. So you're obviously going after things close to my heart with deorphaning, enzymes. So one of the things that'd be cool, I think, for co-ed to do is, do you think there's ways to pair that pipeline with stuff that will take into account the synteny of these enzymes to maybe inform its physiological role, or other ways to pair functional genomics from public databases to at least give us some co-expression? What other types of data can we use to pair with this that will inform it?
That first point is immediately on my to-do list. So think of, for instance, what the Genome Neighborhood tool does, right? Where it looks at the neighboring genes. So right now I'm only looking at two domains that are in the same ORF. But what if it's the next ORF over, or two ORFs over? I think that would be very interesting. It's more computationally intense, but it's certainly doable, I think, because the Genome Neighborhood tool has done something similar to that before. Expression data I haven't thought about yet very much. That's not a bad idea. Maybe I should look into that. But yeah, it's definitely on the drawing board.
And the second part of the second question is, so you have this huge list of enzymes now. What do you think the best way of prioritizing functional work goes? Do you want to go for the super weird stuff, or do you want, so if you have like X number of domains, do you want to try stuff where you have one domain that's known and a couple totally off the wall domains? How do you prioritize study?
I mean, in practice, I prioritize by pet interests.
I'm aware from the Ruber stuff, yeah.
Right, yeah. So you come across a paper and it talks about a reaction done by a domain, and you're like, oh, I wonder what other domains this one comes with, right? And maybe we can find something interesting. But if you're into functional gene annotation, maybe you want to prioritize by the ones that are most common in life that have not been annotated yet, because those are maybe the most important. I mean, just because it's been sequenced a lot doesn't mean that it's actually important ecologically, but maybe it's a decent correlation between there. Other ways to prioritize them. Just making the data public means that a lot of people can look at it and everybody has their own way of prioritizing, and we, you know, there's thousands of enzymes that have not been annotated yet. So I'm really not worried about competition, right? The chance that two people are working on the same enzyme is so small. So I invite everybody to look at these networks and pick their favorite enzyme and go to town, or their favorite domain pair, so to say, and go to town.
Looks like we have a question in the Q&A box from Timothy Fallon. Is it possible to compute co-ed across all of NCBI and make that available?
Yeah, at the moment, I have computed it for all of UniProt. And the nice thing about UniProt is that they have pre-annotated domains. So the PFAM people, every time a new UniProt is released, they calculate the domain occurrences in each of the proteins in there. For NCBI, that hasn't been done for every single genome yet. So it would take quite a lot of computational work, but I think it might be worth investing into that computational work and doing it. Of course, it's going to keep on releasing, so you have to keep up with it a little bit. But at the moment, we've already done it for UniProt, so that's a good start. And there's so much already to discover, right? So I don't think we have to wait until I've done it on NCBI. We can get started already.
Great.
Really great talk, by the way. So can you give me a flavor? This is a bit outside my research area. So how easy is it to predict the function of the genes that you're picking out? So can you quite accurately pick out what it does in the broadest sense, or is it really quite a brute force screening challenge to try and find out what it does?
Yeah, I think, you know, for some, like if you have two domains that are known to be acting in subsequent reactions in a known primary metabolic pathway or something like that. So for instance, you often see gene fusions between two steps in amino acid biosynthesis or in the charismatic pathway. You know, there you can maybe have a good guess as to what it's doing. But in the example that I just showed, the Phi-F and nitroreductase, you know, you just have to go through the literature and see what kinds of reactions they do. And it took me quite a long time to figure out what the products of those enzymes were. And yeah, there's just no shortcut to doing thorough biochemistry and good wet lab work, right? You know, we can get better techniques, we can get better NMRs, we can get better mass specs, but at the end, you know, you have to put in the work to annotate those genes.
All right, thanks.